Doop doo doo doo, it's intro music. This entire structure that you see here, minus that sperm, was ovulated from the ovaries during ovulation. So released from the ovary and picked up by the fimbriae of the uterine tube or fallopian tube. And now it's moving through the fallopian tube, and there it encountered a sperm. This part right here is the secondary oocyte. That's the nucleus of the secondary oocyte, and the yellow part is the cytoplasm. The surrounding white layer is zona pellucida, and it's a non-cellular layer of kind of goo glycoproteins, while the cell layer that is surrounding zona pellucida is called corona radiata, and that was part of the original follicle, and these were the innermost granulosa cells. As this structure is traveling through the uterine tube, it must have encountered many sperm. Only one sperm is shown here. As the acrosome of the sperm is interacting with corona radiata cells, it initiates the oocyte to complete meiosis II. When the sperm encounters the oocyte, the enzymes of the acrosome degrade a path through this layer of cells and zona pellucida so that the head with the nucleus of the sperm can enter the oocyte. This causes a chemical change in the oocyte so that zona pellucida becomes hardened and impermeable to other sperm. This is to prevent polyspermy, or getting nuclei of multiple sperm inside one oocyte. Now we can see the cross-section of the two nuclei, one from the sperm and one from the oocyte, coming together. Meiosis II is finished, and you can see the two polar bodies. So this is a zygote, a single diploid cell that results from the fertilization of an ovum. So this is after the two nuclei, one from sperm and one from the oocyte, have fused. You can see that white polar body over there still. The zygote is a very transitory phase. Very quickly, right after the fusion of the two nuclei, this cell will initiate the first round of mitosis, which will result in two genetically identical diploid cells. These two cells that result from one round of mitosis are totipotent stem cells, meaning that at this point they can develop by mitosis into any cell of the body and any cell of the extra embryonic membranes. After the second round of mitosis, we now have four cells called blastomeres, and they're genetically identical diploid cells. Notice here that the size has not changed. Overall, the zygote, surrounded by zona pellucida, is the same size as the 2 blastomere stage and the 4 blastomere stage. This process of mitosis without cell growth is called cleavage, and it will continue until implantation. As cleavage continues, and there have been 4 rounds of mitotic division, we have a ball of cells, about 16 blastomeres. That ball is called a morula. All of these cells are diploid, they're genetically identical to one another, they're totipotent, and they occupy the same amount of space that a single zygote did. This stage is reached about 4 days after fertilization in humans, and this ball of cells, the morula, is in the uterine cavity but has not made contact with the endometrium yet, and therefore has not begun implantation. This is the same structure on a different model, a morula. Each one of these cells is a blastomere. So each one of those cells is a diploid cell that's genetically identical to all of the other cells. Notice that the entire morula is the same size as the single-celled zygote was. This is all because cleavage does not result in cellular growth, only in cell division. Around the 5th day, the morula, continuing to grow by mitosis, becomes a blastocyst. It is a hollow sphere of cells with a fluid-filled space inside there, the hollow space, called a blastocele. That's the space. The gray cells on the outside are called trophoblast, while the yellow cells on the inside that kind of invaginate in there are called inner cell mass or embryoblast, because they will become the embryo. Around day 6 or 7, the blastocyst grows and makes contact with the endometrium. The inner cell mass starts to grow by mitosis and differentiate. So at this point, cleavage is no longer happening because cells are changing, growing, and differentiating. This is still a blastocyst. The gray cells are trophoblast. The yellow and orange cells are inner cell mass, also called embryoblast. And the hollow space in the middle is still called the blastocele. This model is showing the continuation of that process after the blastocyst touches the endometrium of the uterus. And in this model, you can actually see the endometrium of the uterus. Here it's labeled with numbers 7 and 8. And at this point, endometrium is called decidua. Here we can see the inner cell mass, and it is surrounded by trophoblast cells. The trophoblast cells that encountered the endometrium, or decidua as it's called now, they end up kind of invading the endometrial tissue, and as they grow, their cytoplasms fuse, so they form this large multinucleated mass of cells here in burgundy, labeled number 6. That is called syncytiotrophoblast. Sync as in coming together because the cytoplasms have fused. And as they do that, they degrade the endometrium, and maternal blood kind of pools in these little lacunae. The one layer of cells right there is cytotrophoblast. These cells are not fusing. Same stage of the same structure on a different model. Here, this is the blastocyst that is beginning implantation. All of the gray cells are trophoblast. However, the trophoblast cells right here are specific kind of cells called syncytiotrophoblast because multiple trophoblastic cells are fusing together. This one layer is called the cytotrophoblast, and they're not fusing. These are little lacunae of blood that are forming because the syncytiotrophoblast cells are eating away the endometrium or decidua and breaking the maternal blood vessels, which then leak out into these pools of blood. This part of the inner cell mass will develop into endoderm and ectoderm. As implantation continues and the conceptus grows, it becomes completely enveloped by the endometrium. Embryonic disc forms from the inner cell mass. You can see those two cell layers on this diagram that make up the embryonic disc. Notice that the embryonic disc is surrounded by two cavities. One of the cavities seen here in yellow is the yolk sac. The cells that outline the yolk sac came from the division of the inner cell mass or embryoblast. The human yolk sac does not store yolk like it would in birds and reptiles, but it serves other supportive roles, and some of the cells of the structure do develop into parts of the human. On the other side of the embryonic disc, we see the second cavity and sac called the amnion. Cells right there, labeled number 1, will become most of the embryo and eventually fetus, while the rest of the pink cells, labeled 2, will eventually grow to envelop the whole fetus, becoming the amniotic membrane filled with amniotic fluid in which the entire fetus grows. Surrounding the yolk sac, amnion, and blastocele is this labeled dark burgundy color, is the chorion. The chorion grew from trophoblast cells that were dividing and kind of invading the endometrium, and the chorion becomes the outermost membrane enclosing all of the other membranes and the embryo. At this point, about 2 weeks after fertilization, notice that parts of the chorion kind of has little finger-like projections. Those are called chorionic villi, and they completely surround that blastocyst. The entire conceptus is embedded in decidua or endometrium, labeled 13 here. The same stage on another model, gray being the chorion. On this model, the embryonic disc is forming here. You can also see chorionic villi. Same structure on the model. This is the embryonic disc that's developing into this multicellular structure here. That is the beginning of the embryo. Number 1 there will develop into the head, and that's the tail part of it. You can see that the yellow region is still the yolk sac and the pink is the amnion. Eventually that pink will wrap all the way around the embryo and become the amniotic sac that cushions and protects the fetus. Here, number 10, we can see the well-developed chorionic villi. They are now part of the fetal portion of the placenta. In between them, you see intervillous spaces. These are filled with maternal blood, and gas exchange and exchange of nutrients is happening between the thin layer of cells that surrounds the chorion and the intervillous spaces that contain maternal blood. Here's the embryo and the developing placenta about 4 weeks after fertilization. The burgundy and that dark pink labeled 5 is part of chorion still. Here, number 1 in yellow is the yolk sac that's going to continue diminishing from this point on. The white right here labeled 4 and 3 is the amniotic membrane or the amnion. Inside of it is the amniotic cavity filled with amniotic fluid that bathes the embryo. When people say, oh, my water broke, what they mean is that the amnion broke and the amniotic fluid has spilled out. This is, of course, the developing embryo, and you can already see where the head part is going to be. That's the tail part. Here, the arms are developing. In blue here, we have the oxygen-poor umbilical arteries, so blue in arteries, and a single red umbilical vein that brings highly oxygenated blood from the placenta to the embryo and later fetus. Here, number 2 is the developing umbilical cord, and that includes the vein, arteries, and allantois inside. I also want to point out here that the chorionic villi are really well developed here. The entire burgundy part surrounding the entire conceptus is the chorion, but on the right side, it's going to become really smooth, and those chorionic villi on the right will diminish. On the left, they will become as big as they can be, and in between the chorionic villi on the left there, you can see the intervillous spaces filled with maternal blood. Overall, the placenta is developing on the left
Villi.
side, on the ventral side of the fetus.
Villi.
That's where you're going to have those long chorionic villi and all of the blood exchange. The part of decidua or endometrium that is part of the placenta, so here it's number 7 on the left hand side, the most outer pink layer that you can see, is decidua basalis, because that's where the attachment happens. On the other side, and this is not obvious on this model, on the dorsal side of the embryo, you have decidua capsularis. It becomes more obvious as the fetus grows, but this part of the decidua or endometrium is decidua capsularis, and this part is the decidua basalis. Decidua capsularis encapsulates the fetus and is not part of the placenta. Several structures are visible on this model. In yellow is the right ovary here, broad ligament, dark colors, the fimbriae of the uterine tubes, and then the rest of the uterine tube on the right attaching to the uterus. Here we can see number 9 as the developing umbilical cord and 10 the growing embryo. We can also see on this model that the purple part is the chorion, and where I'm touching right now, that is the developing placenta, part of A and B. We can't see the decidua capsularis here yet, but 13 labels the uterine cavity. This membrane here, white, labeled as 11, outlines the amnion. Inside of it is amniotic cavity, 12, that's filled with amniotic fluid that bathes and protects the growing embryo. As the fetus grows, you can clearly see the umbilical cord that's made up of umbilical arteries, vein, and allantois. 10, of course, is the fetus. 8 is purple, it's chorion, and that's part of the placenta. That red B is also part of the placenta, decidua basalis. Here's the developing fetus at about 3 months. 10 is the fetus. It lies inside of the amniotic cavity, surrounded by the amniotic membrane. The fetus is connected to the placenta by the umbilical cord there. The purple is chorion with chorionic villi. That is part of the placenta that is made up of the fetal tissue. And the dark red there that lies touching it is the decidua basalis, and that is the maternal portion. Here are the chorionic villi with intervillous spaces filled with blood between them.